Okay, we will go ahead and get started. Welcome, everybody. Good morning, good afternoon, good evening. Welcome to the third day of May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. We are very excited that we have this opportunity for all of us to come together and talk about important issues related to proteomics and especially computational, with especially computational focus. And we're very happy that it is possible to do this in this online format despite the circumstances. So if you are joining us for the first time, I hope that you're all healthy and safe. I hope that you can manage the time. And I also hope that these 2 weeks will be a good use of your time. So today is a very special day for us because we have two keynote presentations by leaders of proteomics, and our speakers have made very important contributions to the field. And so while most of the other sessions in the Main Institute are fairly technical, where we work with specific software tools or specific types of experiments, the keynotes are really meant to highlight the applications of proteomics and the applications of computational methods and experimental methods to biological discovery and talk about the use of this technology in the broader context. So we have technically, just from terms of logistics, so as you're logging in, you will see at the bottom of your screen, there are two options. One option is a chat. So chat is useful to talk to presenters or to moderators about if you have a particular question, but administrative question. But if you have a scientific question related to the presentation, it is best to use the Q&A button. And so you can type your question and we will interrupt the speakers and we will voice your questions so that the questions can be answered more or less in real time. If you feel like the questions are somewhat unrelated to the presentation, maybe they will be answered in the end. So please don't hesitate to type your questions, because it is also true for the speakers. It's important for them to know what is relevant for the audience. So this is the plan for today. So let me first introduce our first speaker. It is my great pleasure to introduce Professor Bernhard Kuster. Bernhard joins our program the first time this year, and we are very excited for his contribution. So Bernhard is the chair, he's a professor at the Technical University of Munich and the chair of the Department of Biosciences. So he studied chemistry at the University of Cologne, and then he had this doctorate in biochemistry at the University of Oxford. After that, he was a postdoctoral researcher in Heidelberg and also in Odense. And before joining TUM, he was vice president of CellZone. And the research that Bernhard conducts is in a very broad area of chemical and functional proteomics. And his group is particularly known for large-scale studies which characterize proteomes on a large scale. And we are very excited to hear about large-scale proteomics from Bernhard today. So I'm very much looking forward to your presentation. Thank you very much.
Yeah, thank you very much, Olga. I would have, of course, much preferred to be in Boston on site. I was actually looking forward to this, but we all know that this isn't to be. So I'm still happy, of course, to try this format. And I have to admit, I've never talked to an audience of potentially a few hundred people without seeing anyone except for myself in the camera on the top right-hand corner and Mina and Olga. But I think it's also time to say a big thank you to these two ladies for putting this together and moving it from the analog world into the internet, which achieves two things. Number one, the Main Institute can happen, and number two, it reaches more people than it could otherwise do. So I'm very happy to do this, and Olga was kind enough to provide me with this title, so let's see if those expectations that have been raised with a few people already can be met. However, having said this, I'm going to also structure the presentation in a way that it's going to be quite technical at the beginning, because I do think it's important to really understand what these different technologies can and cannot measure, because that is later on at the heart of our interpretation of the experiments we do, and also the statistics or the informatics that's suitable for analyzing such data may differ depending on what we actually do. So, but before I really get going, I think it's good practice to make some disclosures. Basically, I'm just showing you that I'm a founder and a shareholder of 2 companies, and we do get some research funding from, in this case, Merck, and more research support that is more like in kind from these other companies listed down here. They didn't really have anything to do, well, some of them had something to do with the research here, but not in financial terms, purely academic. Okay, so with that, I would like to sort of pick up speed a little and essentially place TMT quantification within the broader world of quantitative mass spectrometry, because there are actually a lot of choices. And these days, maybe the most popular one are these label-free techniques, either in DIA or DDA mode, or related approaches, where a sample of interest and another sample of interest are basically in the wet lab, processed in parallel all the way down to the data analysis part only, where they're combined at this very last step. And that often times will mean that all of the technical variation that has been incurred up here is going to accumulate and be present in the data. And if there was little variation up here, then analyzing this data is going to be fun. If there was a lot of variation up there, then it's going to be less fun. Now, there's also, of course, the whole world of using standards, and they could be spiked into any sample at the workflow, either at the peptide level. For example, that would be popular for these aqua or PRM or SRM approaches. They can in principle also be spiked in at the protein level. Q-concats are ways of doing this. It's less often used, but certainly possible. What is clearly the case in both of those worlds is that you have to predefine what you're going to look at. So you have to, and you have to make reagents that are often very expensive to make for quantification purposes. And then there is a whole array of possible chemical labeling strategies, where we now talk about stable isotope labeling, and they all have this interesting feature that you can actually introduce labels in a whole proteome, either again on the peptide level by a chemical label, and less frequently by a chemical label on the protein. And you can, of course, also do this already at the metabolic step. The advantage of that being that you can combine your samples of interest at the earliest possible time in that workflow, and then all of the things that may go wrong down that track affect your sample and standard in the same way. So it's not going to really be a problem for the downstream data analysis. Now, important also to keep in mind is that often I get asked which of those many approaches is the best. I basically just tell people, well, depending on what you want to do, choose the one that's fit for purpose, and not necessarily the one that certain colleagues may think is the only thing to do, or that you happen to have in the laboratory. What perhaps still is true is that if you have a very long road from your sample to your data, then these metabolic labeling approaches play out their advantage. If your route from sample to mass spectrometer is short, then label-free is entirely fine. But cardinal differences in many of those techniques is whether the signal that you're going to use for quantification comes from either the MS1 level, so the precursor peptides, or the MS2, so the fragment ions, or maybe MS3, which would also be fragment ions. And there are very large differences between the two, and I'm going to cover this in a little detail. But here on that slide, I'm just giving you some idea of what colleagues have put together, which by no means is a complete list of all the chemical labeling techniques that are out there. And it's also important to note that despite the fact that some of them are quite powerful, including TMT, which I'm going to talk about mostly today, is there are a lot of little devils and a lot of little details in its chemistry, which means it's often not very clean. And so there are side reactions and what have you, and all of that is going to show in your data at some point, and whether or not it's important depends on how bad it is. And now focusing in on tandem mass specs, for those of you who may not have heard much about this, it's a chemical labeling technique, essentially that is by way of this succinimide chemistry able to derivatize the N-terminus of a peptide or the side chain of a lysine residue. And so this is called the NH2 reactive group. Then it has a little linker here, which is also referred to as a mass normalizer, and a reporter group that upon fragmentation by HCD will break this carbon-carbon bond, or by ETD will break this nitrogen-carbon bond. And it comes in a variety of flavors. So by now, it comes in 6, 10, 11, or 16 plex. And what's interesting about it is you can place stable isotopes around that molecule, as depicted down here, in a way that in this case, you have always 5 stable isotope centers for this TMT6 plex in each of those reagents, but just in different positions. That has the effect that the actual compound has the same mass. So we call this isobaric. However, when fragmentation happens along this bond, you can just count the number of blue stars in each of those reagents, and you will find out that there's none here, there's 1 here, there is 2 here, 3 here, 4 here, 5 here. So you can distinguish them in the tandem mass spectrum by the fact that they are all spaced by a certain amount of mass, roughly speaking, 1 Dalton. So, and that is the basis for the quantification. Okay, so, and then there is higher plexing than 6 plex, as I said, 10, 11, and 16. And when you look at the prototypical spectrum of that, you will actually see that each of those channels that has a nominal mass of either 126 or 27, that many of them now come in 2 flavors. There's a split peak here in the 127 channel, 28, 29, and 30. And that split arises from the fact that you can either have a C13 compared to a C12, or a N15 to an N14, and the neutron in that chemical element is actually hasn't got the same mass, but there is about 6 millidalton mass difference. And in order to be able to detect this, you need a mass spectrometer with reasonably high resolution. About 50,000 will baseline resolve those peaks. And that is important to know, of course, when you choose the mass spectrometer for such experiments. Okay, so a typical workflow might then look like this. Important also for TMT experiments, you have to pretty much define a fixed number of experiments you want to compare later on. That's not entirely true, but most of the time people do this, and you could have, say, 6 or 10 or 16 dishes of cell culture, and then you basically want to treat it in 16 different ways. You do that, you harvest your cells, you digest the proteins into peptides, and at the peptide level, you introduce your different TMT labels, then combine all of the samples and do a combined LC-MS-MS analysis, where your MS1 spectrum will have a lot of precursor ions, and then you select out, or the mass spectrometer selects out 1 for MS-MS, and you get your fragment ions. And importantly, now at the low mass end here, you will find your TMT peaks, and they will split up according to the plexing level that you've employed. And then, importantly, the quantification of these 6 or 10 or 16 samples is going to happen relative to each other, and not relative to some spike standard or some such. So it's always a relative quantification. So another cardinal difference between a TMT-based experiment and, for example, using XIC, so extracted ion chromatograms from label-free or DIA or even PRM, is that XICs integrate either the MS1 or the MS2 signal across the chromatographic elution time of a peptide. TMT does not do that. TMT captures the MS2 signal intensity at a certain point in time. And that is largely arbitrary. So we don't know if we're going to, in TMT world, pick the chromatographic elution time at the apex, or early on, or late, or somewhere in the middle. So that's very often not really within our control. And that's an important difference, because we use, in this case, the integrated intensity across the LC peak, and in TMT, we just take what we get at that moment in time. And that has some consequences. Okay, also a word about mass spectrometers, because they are still sort of ordinary detectors, quite powerful, still ordinary detectors. So many things apply that apply to any other detector. First of all, one has to realize that any quantitative analysis tends to need better data than qualitative analysis. But more importantly, detectors all have a certain range within which they can measure properly. If your signal is great, like you have an isotope cluster here of a particular peptide of, something like 1870 Daltons. You see all the isotopes, they are nicely resolved and they follow exactly the isotopic pattern that one would expect. However, if you had very, very high signal, you may actually saturate your detector and then your signal might be squared off. And you can imagine that this would be detrimental for the quantification. Likewise, at the other end of the intensity scale, you might be losing sort of your peak in some, not necessarily only noise, it could just be noise, so when the signal is very low, or there could be other interfering peaks here, which makes picking the right isotope cluster in here not can make it difficult. So we basically need to keep in mind that if you have saturation of your detector, then the quantification is going to be wrong. If you have a too low signal, that may prevent detection, you're going to create missing values. Or if you have a low signal to noise or interfering signals, you get false measurements or false quantification. And what we call dynamic range really is that difference between how little can I detect in the presence of how much of something else, and that will also have an impact on the quality of your quantification. So that's
there's
true.
A question.
Yeah.
Can TMT label be done at the protein level as opposed to peptide level?
Yes, it's possible, but it's very rarely done because when you want to label native protein, you have to do this sort of in an aqueous buffer system. A lot of the residues you may want to label might not be accessible. So you get a lot of variable labeling on different sites. You may actually want to do this because it may tell you something about whether those places on the protein are surface exposed or not. But it's quite rarely done, but it's possible. Okay, so more words about MS1 and MS2 signals, just to keep in mind. Usually MS1 signal intensity, so the precursor, peptide precursor, is usually much higher than the signal in MS2. However, and that you can see from this ion chromatogram up here, these are all the precursors that hit the detector at any one given time in this chromatogram. And if you strain your eyes, you can see that this number is higher than that number. The bottom one is, in this case, a MS2 trace for the ions in all the tandem mass spectra over time. And you can see it's much more spiky. There are and it kind of is cleaner. It's often considered cleaner. It has lower noise or fewer interfering signals. So, but these are important differences that are going to have consequences a little later. So now going into the trenches of TMT, you may or may not know that by any stable isotope label are rarely or never 100% pure. So there are extra peaks in the spectra, both in MS1 and MS2. So if we look at our precursor ion here again, it has that nice cluster of isotopes, but there's also one signal at -1 Dalton. And what that is simply that one of those carbon-13 or nitrogen-15 atoms isn't there. So the purity of the isotope label isn't 100%. And when you buy TMT reagents, the vendor gives you a wash list, how we call it, of how much signal or how little signal you should expect at the -1 position. And then, of course, this is not only true in your MS1 signal, but if you fragment all of this, then this isotope impurity will also show in the MS2 spectrum. And of course, also your TMT reporter ions will also generate a C13 satellite ion, and that has to be corrected for. Now, there is uncertainty in the isotope impurity correction in that list. So that's usually what people don't necessarily think about, that it is actually there. And the accuracy of determination of this isotope purity is actually not much better than 10 or 20%. So when you now take any TMT signal that is uncorrected, here is a channel 126, 127, 28, 29, 30, 31, you can actually see that there is at the -1 position, there is some signal. It's not a lot, it's just 0.2%. But when you then correct it, it should be gone completely, but that doesn't sometimes happen. So sometimes there's residual signal even after the correction. And if you look at the green channel here, that's the uncorrected data, and here's the corrected data, you can see that some of those numbers are now closer to 0, but there are some that are still not 0. And it could be different for each channel. And that has, again, consequences we're going to see in a moment. Yet another thing that most people are probably not aware of, and often it's taken care of by software these days, but also an ion trap detector can be saturated. And people around Markus Banschew have noticed a while ago, or it's already a few years ago, that if you put too many ions into a C-trap or an Orbitrap instrument, then you get space charging, and that can lead to what is called ion coalescence. So here we're looking at the heavy and the light channel of the 130 TMT reporter ion, but if there's too many ions there, they can coalesce into a single peak, which means this quantification of this peak isn't somehow representation of those two, but there's no way of knowing whether this is a 1-to-1 mix or whether it was a 9-to-10 mix or a 1-to-10 mix. We don't know. And that could happen to various degrees in the different channels. And one tries to overcome this or prevent this by making sure that one never allows too many ions into the system. Okay, so let's say we have all of this under control. You can get some idea of what the dynamic range of your quantification may be. And here we did a couple of experiments a while ago where we just took HeLa digests and diluted them separately and then started to mix them together in various ratios. Because we always use the same digest and we dilute them, we kind of wouldn't expect any sort of what is called ratio compression. I'll come to that in a moment, because every peptide is diluted by the same factor. Now, but if we do this, and we now look at this plot down here, what you can see in this plot is that when you mix TMT channels together that have different quantities of digest in them, you can calculate, of course, what you would expect in terms of ratio, and this is in a log 10 scale, and then you can measure what you actually get in the experiment. And in this case, when we just look at the 200 most abundant peptides, you can see that while we're diluting the sample, the signal goes down, and your expectation, the expect ratio of the 1-to-1 versus 1-to in 1,000 mixture is actually quite good. So this goes linearly down. But then when you go to about 1 in 1,000, you hit the bottom. So which means when you dilute the sample further, you don't further decrease your signal. Okay, so clearly we've reached the end of the dynamic range here. Now, you can do a second experiment where we now keep every other TMT channel empty, and again, analyze the 200 most abundant peptides. And then you see something perhaps surprising, that while the normal dilution channels that actually have digest follow exactly your expectation of going down and down and down, with the expected and the observed ratios being the same. However, in the empty channels where we didn't even put any peptides in, we begin to see signal. And this could be as much as 1 in 100 or down to 1 in 1,000. So our effective dynamic range that we can actually measure is decreased to 1 in 100 or to 1 in 1,000. Now, and if we now take all TMT channels and occupy them with sample and dilute, and now we can look at, let's say, the top 10 most abundant peptides in that sample. They again are nicely behaved along this axis, but you'll quickly see that even when you look at the top 100 peptides or the top 1,000 or all of them, you can see that we are populating here signal in dilutions that we really shouldn't have any more signal. And it could be not as bad for the intense peaks. It's still 1 in 1,000 dynamic range, perhaps. That's the green dots. But it could be as bad as 1 in 20 for the less abundant peaks, like on this blue thing here. And if you look at the whisker up here, this is not much more than 20-fold, even though you would expect that to be a 1,000-fold. So, and then similar argument applies to that if you actually hit 0 and have missing values in your TMT experiment simply because there wasn't enough signal, then you can, of course, impute values. But if you do that, you have to be aware of the fact that if you impute values for peaks that are usually very intense, again, near the top 200 peptides in these dilution experiments, this is fine. You can impute, but then you still hit a bottom at some point, and that bottom that you hit is actually quite close to the 1 in 1,000 we had over here. But if you look at the least abundant peptides, then you're hitting that already at a factor 1 to 10. So all of that means is your quantification results are also going to be influenced by what your detection system can actually measure. So be mindful of that fact when you look at TMT data. Sometimes it's not a problem. Sometimes it's a terrible mistake.
Bernhard, could we take a few questions? There are quite a few.
I imagine, yeah.
I think we will not be able to answer all of them, but let's just do a few. So can you comment on label-free versus TMT in terms of dynamic range?
Yeah, that'll come in a few slides, if I can hold that off for a moment.
Sure thing. And so then how can we reduce or avoid reporter ion interference?
Yep, that's also it's a very good question. And one easy way of doing this is by adding another step of MS, which is called MS3. So you're selecting your fragment ions again in your trap, and then you're fragmenting those fragment ions to create reporter ions that you then measure sort of in an MS3 level experiment, and you can reduce much of that interference.
Another question, what is the usual amount of ions safe to use to avoid space charging in orbit traps?
Yeah, so
orbit trap size is determining the amount as well.
Yeah, well, the capacity of the trap is determined by many things, but if you want to play it safe, 1E5 ions is what you can put in usually.
Another question is, if you have an empty channel and if they're not always to be zero, how to characterize the labeling efficiency, and what kind of percentage or intensity is tolerable? Well,
the labeling efficiency is actually usually best evaluated either at the MS1 level or actually at the level of a search engine. When you go searching with TMT labels either as a variable modification, and then you can just count the number of peptides that only have 1 or 0 labels, and you can look up their intensities and build essentially the ratio of the intensities with a label and without a label, and that gives you a broad sense of the labeling efficiency.
And maybe the last for now, what software do you use for MS3 level analysis?
Most of the time we use MaxQuant.
Okay, thank you.
Okay, so after all this, we're going to look at one other thing, a term that many of you will have heard about, which is called ratio compression. The examples I showed you previously ideally wouldn't have, as I said, any ratio compression because it's all the same peptides, just diluted at the same relative ratio. So we shouldn't see any distortion in those ratios. However, in practical terms, there is this phenomenon called ratio compression or ratio distortion, and this really arises from the presence of other peptides at the moment the mass spectrometer decides to pick one precursor for fragmentation. So we can take a look, for example, here you have this black isotope cluster here for one particular peptide, and there's some red signal here, but it's at a very low level. And if you then do MS, your reporter ions are going to hopefully mainly represent the black ions here, and the red ions might be representing a smaller amount of the signal. So the contribution of the red ions to the black signal isn't going to be very high. So therefore, we can expect that the ratios that we're going to compute from one TMT channel to the other are going to be relatively close to what the actual ratios are. However, just a word of caution, we of course have absolutely no way of knowing if these interfering peaks are going to produce, like I depicted here, a 1-to-1-to-1-to-1 ratio, or if they themselves have ratios in that experiment. But as a simple sort of matter of principle, if you have low interfering signals, you usually get little ratio compression because the contribution of that interfering ions here to the total TMT signal here isn't very high. However, if you have more interference, and it happens more often than you think, then you could actually have a much bigger contribution. And again, if we assume that the red ions here are all 1-to-1-to-1, you can actually see what you measure is the total signal, but your actual ratios of your black things would have been much bigger than what you can actually measure. And this is why it's called compression, because the true ratios are always appear much smaller than they truly are. And this would lead to actually quite a bad accuracy. And accuracy being defined how close is my measurement to the actual true value, whereas precision just refers to how reproducible my measurement is, no matter how correct it is. So, and on the right-hand side, and where this really shows is when you start mixing different things together. So in this experiment, for example, we took, I think, 10 or 11 cell lines, digested them, labeled them with, where's my mouse? My mouse is gone. Labeled them with TMT, and then ran them down different types of LCMS. And we then ask the question in this plot is how many of those peptides show kind of what range of TMT quantification, and that's a log2 scale here. And you can see that no matter what we do, the median or the sort of the majority of all those TMT reporter ions have ratios that are not much bigger than 3 in log2 world. So that's a factor 8 or maybe 4. That would be a factor 16. Some of them extend all the way up to a log2 of 10, which is roughly 1,000-fold, but only very, very few. And to the question that just got asked, so what can we do? So if you look at the 4 lines, the first one being a nano LC and MS2 readout with a short gradient, then our range of dynamic range in which 95% of all data is just 1 in 50. We can do a better chromatography, then it may just increase to 1 in 75 or so, simply because not as many peaks are eluting at the same time. But we can then also do the MS3 experiment that I alluded to, and then it goes up to 1 in 100, and we can then extend again the gradient for the LC to simplify the mixture, and then we can go up to 150 in this range. And here, mind you, this is all exactly the same sample. Okay, but your quantitative dynamic range is quite different depending on how you analyze it on the mass spectrometer. So that's important to keep in mind. Okay, so for some reason I cannot get my mouse back. Can I have a quick?
It is back.
You can see it. Okay, yeah. Very good. Okay,
and
so
maybe can we do a couple more questions as well? So yeah. Ratio compression may be high for one peptide, but low for another peptide. Is there a method to correct for quote unquote noisy peptide?
Yeah, so there are approaches to do that. I mean, there are some software tools that can actually look at that window that got isolated and then ask how much of the total signal in that window can we attribute to the black or the red signal. And then this is applied to correct this. However, it works under the assumption that most peptides are not changing in an experiment so that they would produce 1-to-1-to-1-to-1 ratios. And then this is a way of how one can clean this up. But as I said, it is an assumption that may or may not be always justified. But generally, this works, and it proves matters. And mind you, in the plot on the right-hand side, we've already done that.
And another question is, what is a good cutoff for interference?
I don't think there is really such a thing as a good cutoff, because it is a continuous scale. It's hard to know. What we generally observe is the more peptides for a protein we have, the better the quantification becomes, because you can either exclude the weird ratios completely, or if they have, let's say, not much intensity, then they also don't contribute so much to the total signal. But there's really, it's hard to deal with this post data acquisition. A very new way of dealing with this has been published also by Steve Giggy's lab, where you're on the fly trying to identify a peptide from MS2, and then you only do MS3 on those peptides that you've already identified, and then that actually produces very good results in terms of removing ratio compression issues.
So a couple of technology questions. Why high flow rate is giving better dynamic range than nano?
Yeah, so if you go to higher flow rate, we just do better chromatography. For those of you who look at your nano LCMS chromatograms, sometimes you're shocked and sometimes you're in awe. Often, essentially, these very narrow columns, 50 or 75 micron, they don't have a lot of capacity. So even the slightest overloading of the column will produce broader peaks. And broader peaks in chromatography means more precursors present at any one time. And if you go to higher flow rates and bigger column diameters, simply your chromatographic peak width gets smaller and smaller, so you're reducing the complexity of precursor ions that fly into the mass spectrometer at any one time.
And just two more technology questions. With low sample volumes, is high flow LC possible?
With low sample volumes or quantity, you mean? Maybe quantity is what is meant.
Probably.
Yeah, so higher flow rates and bigger columns usually eat some signal. At the same time, the chromatographic peaks tend to get much sharper, so the concentration goes up. And because electrospray is a concentration-dependent process, some of it can be compensated, but only within limits. So the highest sensitivity will be achieved in nano LC, and when you go to higher flows, you will compromise some sensitivity. But what people are observing when they go from 300 nanoliters to 3 microliters a minute, they don't really lose a factor 10, which you should in theory lose, but they only use maybe a factor 1.5 to 2. And when you go to much higher flow rates, even like 100 times higher than the typical nano, what we observe in our lab is that our sensitivity only goes down by about a factor 5 to 10. And for many proteomic applications, this is not a problem. For some proteomic applications, it's a complete killer and you can't do it.
And the last question for now. So can you comment on HPLC versus ion mobility fractionation?
Yeah, well, that's an early, let's say an early field. Of course, I would never compromise LC quality for anything if possible, but if you have to, then ion mobility can compensate to some degree. What we find is that if you call it if one would want to call it the peak capacity of ion mobility is smaller than that of chromatography. So therefore usually gain actually faster if you do better chromatography, but that may depend also whether you are dealing with a Brucker times type of ion mobility or Waters type of ion mobility or thermo type of ion mobility.
Okay, thank you. There's a lot more questions, but
Yeah,
let's move on and maybe we can answer towards the end.
Yeah, so there's a reason why I'm dwelling so much on the techie things, because TMT, we like TMT a lot and other people like TMT a lot, but it has some sort of nasty side effects. And one is shown here. When we look at tandem mass tech use and reproducibility, what this ratio compression issue can do and will do is it leads to a great apparent reproducibility between your experiments. So let's say you have done a TMT 10-plex experiment and you have triplicates of 3 experiments in here to occupy 9 of your channels. You can, of course, ask the question, how do my TMT channel intensities correlate with each other? And if you have replicates, you can ask the question, how do the replicates correlate to each other? And then if you get a very high number, you may be happy because you say, I'm very reproducible at what I'm doing. And this is probably interesting for the stats people. So if we look at this particular example from a TMT 10-plex experiment where we only used 9 channels, but here's 3 replicates that you can correlate to each other. Each replicate also had 2 different conditions. That doesn't matter for now, but look at these Pearson correlations between those experiments. They are near perfect. And if you use coefficients of variation for this and plot them out, then it turns out that in this data set, on the level of phosphorylation sites, we get 12% CV on 10 or 20,000 phosphorylation sites. That's pretty good going. That's better than any PRM I know. Or if you roll up the TMT intensities of peptides into proteins, the median CVs are down to 2%. So much of that is driven by ratio compression, because if your signal is compressed to 1 to 1 to 1 most of the time, then obviously you're going to get a very great apparent reproducibility, but please don't interpret that such that TMT has great quantitative precision. And that is what I see a lot in the field, that TMT is sort of equals great precision, and to some degree that is true, but a lot of that precision is only apparently so because of ratio compression. Okay, so the question was asked, so how do the 2 levels, MS2 and MS1 or MS3 and TMT and label-free compare? This is what is shown in these plots. And one thing, again, to keep in mind is that MS1 peptide detection is actually most of the time limited by signal to noise. There's usually a lot of signal, but if your noise level is high, your signal to noise is low, and then you may not be able to quantify well. MS2 or MS3 peptide detection or fragment ion detection is usually limited by the actual signal. Do you have enough signal? Then you get nice measurements. Don't you have enough signal? You get poor measurements. It's important to remember that both of them have a finite dynamic range. And this is what is shown here on this plot. Here, this distribution is TMT reporter ion intensity on this axis. And you can see it spans about 4 orders of magnitude if we look at across all LCMS runs, I should say. And here the equivalent actually label-free, it's on the very same data. You can see it has this distribution, and these 2 distributions are quite strong, far apart from each other. roughly by a factor of 1,000. And that reflects the amount of signal that is in your detector. And you can see another thing is that here on the label-free MS1 dynamic range, you can see that this side sort of looks like a nice right side of a normal distribution, and then it sort of crashes down on this level. This is where the background signal kick in, not necessarily noise in terms of electronic noise because they have very little of it, but other stuff that is eating the dynamic range of your detector. In MS2 or MS3, you have a lot less signal in total, so you have a lot less problems with signal to noise issues, and there the distribution is actually a bit smoother on both sides, or more symmetrical. So what that means is some of your quantification results in MS1, if they are down here, could actually be distorted, whereas that shouldn't happen so much at the level of TMT in MS2 or MS3. Now, if we roll that from the MS intensity to the peptide dynamic range, we can actually see that for TMT and label-free, it's actually not so different. And the median is actually at about a factor 32 for label-free or 37 for TMT, so not really much difference. But if we now roll up the peptide intensity information into a protein quantification, then we see that the label-free has a bit of a larger dynamic range than the TMT, and that is simply because we can add up the ion intensities of MS1 signal in a way more efficiently than we can do that with TMTs. So peptide level quantification is just as TMT, just as good as MS1. Protein level has a bigger dynamic range in MS1. So another point that is very often discussed, and I therefore want to cover this as well, is the missing value issue. One of my favorite topics. When we do single TMT experiments, we observe that we have typically very few missing values. But I would argue, or I would like you to think about whether they are not missing for the right or the wrong reasons. And what you see in this plot down here is actually 55 lung cancer cell lines that we've analyzed, either by label-free quantification, that's the left sort of block, or by TMT. And we had to use some 14 or so TMT experiments in order to cover all 55 cell lines. And then were label-free, of course, we could analyze them each separately. And when we look at the number of peptides or proteins we observe in the label-free, we get in total about 9,000. In the TMT, we get a total of about 9,000. But if we ask the question, how many of those proteins are actually detected in each and every single cell line, then the label-free comes back with about 3,500, and the TMT comes back with about 6,000. And the green bars are just those proteins that are detected in 9 to 54 cell lines, and the blue that you can probably barely see is that these are proteins that are very rarely detected. Now, from this one might conclude that TMT doesn't get in total more protein in a single experiment, or even when we put all experiments together, but it seems there seem to be much less missing values in the data. But if you now think back to what I told you about ratio compression, this may have a different interpretation. So at some point, the ratio compression that we have in the data may falsely suggest that we have a protein identified, whereas it is actually not present. So how can we get an idea about whether this is true or not? Here are two plots that are essentially showing the same data. On the left-hand side, essentially on the x-axis, we plot the difference or the pairwise comparison of LFQ intensities of a protein in any two of those 55 cell lines. And on the y-axis, we do the same thing for the TMT channels. And there's already two things become apparent. If we look at the x-axis, the dynamic range of the LFQ is bigger. It goes from -6 to +6 in log 10 world, and the axis on the y-axis go from -4 to +4, still a very wide dynamic range. But we can also see that the correlation is not following a slope of 1. So something is different, and it's maybe easier to see on the right-hand side of the plot when we bin now those pairwise comparison differences in the LFQ world on the x-axis and plot them against the actual fold difference. And then we can see that the gray boxes are all those proteins that are within a certain bin. And you can see they follow this linear trend. And in blue are the same proteins, but in the TMT world. And you can see that they have a very different slope. And they also have many, many more, sort of much more spread around the medians. And this is purely a result of ratio compression. And therefore, the reduced number of missing values in TMT experiment actually primarily are a result of ratio compression and not of actual measurement. That's important to understand, and it can be getting very bad. I unfortunately couldn't quickly find the example that I wanted to show, but imagine you have an experiment where you have a protein in one sample and you have a knockout in the other cell line. The ratio should be infinite, and we've done such experiments where we compared knockout cell lines versus wild type, and in the most extreme case that we encountered, the knockout cell line only showed a reduction of protein intensity for the knockout gene by 50%. And the other 50% of the signal had nothing to do with this peptide or protein. It had all to do with ratio compression. So it can get very bad.
Bernhard, could you take a few questions? There is a lot of questions already.
So
one question is it recommended to compare TMT and LFQ as a quality control check on a regular basis?
Yeah, that really depends on what you compare it. If you compare it really on very complex samples, then the comparison always says the correlation is there, but it's not particularly good. Again, that may not mean much for your interpretation of your results. It may just mean whether you're looking at accuracy or precision or what is more accurate than the other. If you, however, use very simple mixtures to ask that question, it should be fine.
So a clarification question, the data you were showing, are these MS3?
The TMT data that we are showing here is actually MS3, yes.
Okay. Another question, how does TMT perform on highly complex samples with large dynamic range, for example, plasma? Is it a large improvement compared to LFQ?
No, it's actually a deprovement, if you like, because the ratio compression problem is going to kick in big time. So I'm going to come to that. I would not really recommend using the TMT for body fluids.
Interesting. Another question, when you have multiple TMT sets for different cell lines, do you search the data all together in MaxQuant? And how do you correct using the pooled sample?
Yeah, so I would recommend searching them together, because only then can MaxQuant make sure that the protein grouping works. So if you search them separately, that is one way of getting more proteins, but that may just falsely happen because you want to make sure that all your peptide grouping, protein grouping procedures are not impacted.
Another question, can the reduced missing values in TMT be the result of using a reference standard sample included in each TMT plex?
That's one of my favorites. I didn't mention it yet, but all of what I said, of course, is highly important for the single cell guys and girls. Because if you're using a reference spike in, if you will, or channel, then depending on how much of that you spike, this is what you're going to measure. And you're going to measure all the other channels relative to that. And if that one channel that you use as a reference has 2 times, 4 times, 10 times, 100 times more sample, then most of what you're going to measure in the other channels, or more and more of what you're measuring in the other channels, is going to just be spillover from the reference channel. So that is a difficulty. But yes, when you have to combine many TMT experiments into a larger analysis, then you should actually use one, better 2 reference channels.
And one more. In TMT experiments, quantifying more PSM per peptide and protein, does it lead to better quantification at the protein level?
It does, but not quite as strongly as for label-free. So it's actually interesting how stable TMT peptide level quantification can be, and that depends a bit on, and really that depends on how much sort of ratio compression you have and how much signal you have. But it's actually interesting to note that the improvement with TMT, or it saturates rather quickly, or it stabilizes, I should say, it stabilizes rather quickly. If you have 3, 4, 5 peptides in a TMT, your quantification usually stabilizes very quickly.
And maybe the last question for now, do you think a narrow isolation window, such as 0.5 m/z, would help to minimize the ratio compression?
It certainly helps. People have shown that. It minimizes, or it makes the problem smaller, but it doesn't remove it completely. Also, when you do MS3, people have experienced that it makes a difference whether you pick 10 of your MS2 fragments for further fragmentation by MS3 or 1. So the least ratio compression you get is actually when you just pick a single fragment ion from your MS2 and do MS3 on it. But of course, the cost of that is very apparent. It is going to cost sensitivity. So in the end, you're going to want to operate a mix of good LC, narrow isolation, and if you can, the computational sort of cleanup of the data.
And maybe the very last one in this batch, do you need any change for a 16-plex TMT, change in the protocols?
In the labeling protocol, not, but what you sort of have to keep in mind is that the capacity of your LC-MS system hasn't become bigger, but you're now pooling 16 samples together instead of, let's say, 6. So all of the available measurement capacity is going to be taken up by all of those ions. So you might actually expect that the minor components in your mixture may be harder to detect, simply because the major components are eating up all of the available space.
Okay, thanks a lot.
Okay, so this is sort of the end of my technical part, and I hope we've been already using a lot of time on questions, because otherwise I'm doing very badly on the large scale stuff. But at least some of the data that I showed you is actually based on pretty large data sets that allowed us to sort of have a statistically sound evaluation of such data. But if you want to at least take all of that with sort of the disclaimer that these are the things that I think, and not necessarily others. But as I said, we actually quite like TMT labeling, and there's a couple of clear advantages. And one is, I haven't talked about this, but you can apply it to any and all biological samples you can think of, at least in principle. And you can multiplex. So measuring 16 things in one go clearly has a lot going for it. And interestingly, and pragmatically, the quantification results are rather less dependent on your LC performance than label-free experiments. So if you're into PRMs, for example, you really want to make sure that your LC performs the same day in, day out. If you do TMT, that doesn't matter so much. This is, by the way, one of the reasons why some PIs prefer using TMT labels even to measure patient cohorts, for example, because they don't have to worry so much about the performance of the LC. Now, we talked about no missing values in inverted commas. So TMT does have, in fact, reasonably good precision. And well, the tag also, to some degree, makes the peptides fly a little better. The downsides of the technology we've also looked at is co-fragmentation can compress ratios, leading to underestimation of the true values. So that means poorer accuracy. And the limited signal in MS2 may compromise sensitivity, and we usually in TMT, because of multiplexing, see a little less IDs than in label-free, but that is not really a big problem. And the last point I alluded to is TMT labeling happens relatively late in a workflow, or oftentimes, or not always, but sometimes. So all of the technical variation that is accumulated up to that point will be in the data and hard to remove. So when should we use what? Now this really becomes entirely subjective. This is why my first bullet is really don't be too dogmatic about it. Think about what is important for the experiment that you do, because sometimes quantitative accuracy is more important than precision, or the other way around. A lot of times you can completely get away with good precision, not worrying too much about whether your quantitative results are actually true. We think it's a great tool for multiplexing experiments that happen within the same biological system, where often, not always, where often precision suffices. So if you run time courses or differential treatment on the same cells, or biological systems. Or protein turnover, I may get a chance to talk a little about this, or this thermal proteome profiling that many labs are fond of doing these days. Where I think it is more challenging, but by no means can be done, but it's just more challenging, is when you're multiplexing samples that come from very different biological backgrounds. Because I would argue you often need the accuracy to really reflect in your quantitative data those differences in biological systems. So panels of cell lines in tissues are a bit of a tricky business, or cohorts of patients, and that question got asked, or anything really where you have to expect very large changes, because very large changes are often harder to analyze by TMT. And what do I mean by large changes? Anything bigger than a factor 20 or 50 or even 100. Okay, so that was the techie part of it. And now I was coming to a few examples of where we're using it. And Olga, you need to sort of tell me how much or how little time I have. I think 50 minutes of our 90-minute slots are up, correct?
So I think you can have 35 minutes more.
All right. Anyhow, so you basically stop me. So the first example where we used TMT a lot is thermal stability of proteome profiling. And one question as a dad of 3 kids has always sort of kept me wondering when there was another cold that came along with fever, is why do humans actually die if our core body temperature rises above 42 degrees? And some time ago, a postdoc in my lab did a brave Bradford assay experiment, basically taking some lysate of cells and just heat them to temperatures between 30 degrees and 100 degrees, and each data point here is one of those experiments. And you can see that when we look at the y-axis is the amount of soluble protein after heating, is that it follows this sigmoidal behavior where starting at about 42 degrees, we are seeing a rapid loss of protein due to precipitation. And this is when essentially, if that happens inside your cells and proteins begin to precipitate at a massive scale, then those cells are certainly going to not be happy. And actually, hyperthermia is characterized by multi-organ failure because the proteins inside the cells are actually crashing. But it's also interesting to see at the other end of the distribution, at the high temperature range, there's about 20% or 15% of the proteins that at least remain soluble. They may be completely denatured, but they don't crash out of solutions. Okay, so this seems as a characteristic of proteins, that they can aggregate or co-aggregate with other proteins when heated. And we set out on an experiment where we asked the question, what actually determines this thermal stability of proteins? And in order to do this, we did a reasonably large experiment, essentially where we looked at 13 different species and heated them up to different temperatures, and basically leading to protein precipitation to various degrees. And we encoded each of the temperatures with a TMT label. And then using the TMT signal, we would basically build melting curves for all of those proteins, for all of those species. And in total, we did about 77 of these experiments, and covering 140,000 such curves, and representing about 48,000 different proteins. And we hoped with that data set, we might actually learn something about that question, what determines protein stability. And here are just examples for these melting curves. And here TMT really was crucial for us, because we encoded every temperature with one TMT label to be able to create these curves with good quality. And you can see here that this protein IK red shows a melting characteristics where half of it has precipitated at around 46 degrees already, whereas this other protein printed in black called MAPT or tau, it doesn't really melt at all. It may well denature, but it doesn't crash out of solution. And so one thing that we clearly learned very quickly is that the melting characteristics of these proteins can be very different from one to the other. And we just, for practical reasons, classified those proteins into what we call low melting points. So where the area under the curve of these melting plots that we have at the bottom here, if the area under that curve was less than 35%, we call it a low TM, so an early melter, if you like, or if it was between 0.35 and 0.65, it was a medium melter. High melters would be characterized by an area under the curve bigger than 0.65, and there would be no melting, non-melting proteins that actually never reach 50% reduction, like here in this case, this tau protein. So that's just a pragmatic way of categorizing the proteins. It's rather arbitrary, but that's what we did to try and ask a couple of questions. And that's going to be, we're coming back to this in a few moments. But one of the things we saw is we actually summarized in this plot when we look at all these different organisms, and they are now organized by the actual optimal growth temperature of them. So at the top is this bacterium that loves living at about 70 to 80 degrees near thermal vents in the ocean, and all the way down to this bacterium, which is called something Antarctica, which is found in the waters of Antarctica actually, and is happy down to 4 degrees, but optimally grows at something like 15 degrees. And you can see that all those organisms show some distribution of the melting points of their proteins and commensurate with the temperatures at which they live. Obviously, the Thermophilus has to find a way to make functional proteins at 70 degrees, otherwise it couldn't live there. Likewise, at the other end of the spectrum, that other bacterium that likes cold water apparently doesn't have to make particularly cold adapted proteins because when you look at the distribution, it's not really much different to C. elegans that lives at 20 degrees, or even Arabidopsis thaliana that lives at about 25, or the zebrafish that lives at about 30 degrees. So one thing that is noteworthy in this data is the open circles. The open circles denote the optimum growth temperature. And what you may be able to see is that at least all of the higher organisms, the Arabidopsis, also the C. elegans, the Arabidopsis, the zebrafish, the fly, humans, mouse, they all have a gap between the optimal growth temperature and the left side, so that's the cold side of the melting point distribution. And we think this is, whereas the bacteria don't have that. The optimal growth temperature is very close to the beginning of where proteins actually start melting. And we think that has something to say about the biology. So these higher organisms have to operate some safety margin because they live in environments that have changing temperatures. But interestingly also that zebrafish, it lives in warm waters, and it's sort of a endless water bath and constant temperature. So it has a reasonably narrow range of melting points because it doesn't really have to optimize this. Whereas the bacteria seem to be all sitting in a niche where they've adapted to these temperatures, but then the moment you take them out of their temperature, particularly on the higher temperature side, they don't like that at all and actually die. So we thought that was an interesting observation in the data. And then we did a series, what I still think is reasonably elegant experiments actually, and asked whether the thermal stability of proteins is an intrinsic property of a protein. So what you see here is a series of 3 experiments where we compared the melting behavior of proteins in this thermophile, Thermophilus versus E. coli. And on the left-hand side, you can see that if you prepare lysates, or if you heat up cells, these are the scatter plots that we plot, you can see that the E. coli proteins on average have melting points of some 50 degrees in lysates and 52 degrees in cells. And the Thermophilus has the equivalent of about 85 degrees. So far, so good. And then we asked, okay, what if we mix those lysates together? So we prepared separate lysates of these bacteria, then mixed them together, and then heated the proteins as a mix. And when we then look at the correlation plots of whether those proteins, of what the melting points are, if either in the individual lysates or in the mixed lysates, we actually see that these proteins from E. coli don't really care much about the presence of the proteins from Thermophilus or vice versa, indicating that just the mere presence of other proteins doesn't really play a role. And then we took about 70 genes from Thermophilus and expressed them in E. coli, so that the host organism now was in both cases E. coli. And then we compared, again, the melting temperatures in cells And then we found out that no matter what we did, the Thermophilus proteins always had high melting points and the E. coli proteins always had low melting points. So from that, we concluded that the thermal stability of these proteins is primarily a protein intrinsic property. Well, one would have maybe guessed this from the start. But then when we looked at this a little more closely, we also found that the environment in which these proteins are also does play a role. So here what you see is maybe 24 cellular proteins that we also happen to find in a couple of human body fluids. And the open circles are the melting temperatures or the AUC, area under the curve values in the cell lines in either HEC293s or in hepatocytes. And you can see that in the colored dots, the AUC values were always much higher in those body fluids. And the interpretation of that is twofold. Number one is you're actually diluting, compared to a cell line, you're diluting out protein, so it can't really find other proteins to precipitate. But it also says that the protein concentration in the cell, in this body fluids are also generally lower than that in the cells, so that it is harder for those proteins to find others to co-precipitate. So that's, I think, an important learning that comes out of this, because it, sorry, I have to stop my timer here, because it says no protein sort of aggregates and crashes out of solution alone. It needs other molecules around it that have a similar behavior. And where this is very clearly the case is when you look at the composition of protein complexes. And on the right-hand side, there is a protein complex from a zebrafish, and each of those yellow lines is a subunit of that complex, and you can see that they all co-aggregate and then crash out together. And then the equivalent complex on the human has a few more subunits, but it essentially does the same. So it appears that protein complexes, this is also what other people have seen, of course, before, but it seems that this is also kind of conserved even from one species to the other. Again, noting the important biological importance of protein-protein interactions and regulating cellular activities. All right, so then on our sort of voyage of trying to understand what the thermal stability of proteins may be driven by, we looked at a lot of things, and it became rather frustrating because even though we found a lot of associations, a lot of them were really, despite being statistically significant, not particularly telling. So on the left-hand side here is a plot where we asked the question about, in our 4 categories of melters, what about protein length? Because it had been suggested that the bigger proteins are more thermal labile. And statistically, we can see that. I mean, if you go from the black to the red to the gray to the blue distribution here, you can see that that the average sort of length gets higher and higher. And it has some ridiculous p-value. However, really, when you see how broad these distributions are, you can't really conclude from that, oh, if a protein is big, it will be thermally not very stable. Now, we can take a little more refined look at this because when we now look at all of the species in this plot in the middle, and again, now this time map out the length of the protein in terms of numbers of amino acids and plot it against the mean melting point of each species, then actually we do see that while the eukaryotes are all over the place, the bacterial species, they do at least follow some trend. And I acknowledge that the statisticians are going to get shivers when you make correlation plots that only have 6 data points in them, but there is some correlation. And there's actually some biology behind this because bacteria have very few ways of making complex functional proteins other than making them bigger. And it seems that Antarctica and Subtilis are a little better at doing this, or if you turn that around, is that if you want to live at very high temperatures, you have to live with the fact that you need to make shorter proteins.
A couple of questions, could we do that? What was the difference in the heat tolerance among homologous proteins between Thermophilus and E. coli?
Yeah, good, very good question. Gigantic. It's pretty much the same distance than for all others. So if we look at the very same protein, or it's actually not as easy to trace all the way from Thermophilus to Antarctica what the same protein is, but we managed to do this for a couple of 100 cases, and we saw the same thing. So even a functionally homologous protein is vastly different in thermal behavior, but if you are that distant in evolution, actually also the identity level on the amino acid level is also not very high anymore, even as little as 20%. So you can conserve a certain protein function, even though the level of amino acid identity may be as little as 20%.
Did you observe a correlation between half-life of proteins versus thermal stability?
Coming in a second.
And one more. Were there more cysteines in the thermophilus proteins as compared to the E. coli proteins to explain the increase in thermostability?
There were actually less cysteines, because this beast lives in thermal vents and then is such a reducing environment that building cysteine bridges won't work. Too much reducing power around.
Does any particular amino acid contribute to high stability?
We'll come to that in the next slide.
And maybe the last question in this batch. In the experiment where you mixed lysate from E. coli and thermophilus, the figure that you showed, was it for organism-specific proteins? Were there any common proteins between those organisms?
That I can't answer for sure, but I would assume that there are functionally common proteins in there, because our chance of detecting something is if they're more highly abundant, and so we're assuming that they're more like housekeeping proteins, but I would have to look that up.
Okay, thank you.
Okay, on the right-hand side, just to finish this, is we can also see some trend in protein abundance, that the higher abundant a protein is, the higher the melting point it also is. Again, statistically superbly significant, but if you look at the boxes and whiskers, I'd say there's very little predictive power in protein abundance too. That may still mean something, but it's not easy to interpret. Now, okay, so as I said, that we looked at a lot of things that turned out to be very frustrating. And in the supplement of that paper, you can actually look at these frequency plots that I show here for every single amino acid in the alphabet of proteins. And we actually mostly saw nothing on an individual amino acid basis. However, when we group some amino acids together, like polar amino acids, particularly those 5 here, we actually did see that those thermophiles had a lot less of these polar amino acids compared to a mesophile like E. coli. And when we then turn this sort of around and ask the similar question, what about the hydrophobic amino acids then? The difference isn't quite as pronounced, but it's quite clear that Thermophilus makes more use of hydrophobic amino acids. And that the interpretation of that is actually in this heated environment, you don't want too many of those polar amino acids around, because the water shells and so can actually disrupt the structure of your proteins. Whereas if you're using more hydrophobic amino acids, you're generating a more compact structure, which goes along potentially then with higher thermal stability. But on an individual amino acid level, we actually saw virtually nothing. And also, the same when we looked at secondary structure elements like alpha helices or beta strands, and here are all of our species, you don't really notice a profound difference between them. So, and if we then go on and more specifically ask the question, what about disordered structure then? One thing that we did see is that the higher organisms all have a lot more disordered structure than the bacteria. Well, that is something that people had known long before. But when we then again look at our 4 categories, in this case in human, we find if we plot that again along the axis of percentage of disordered structure, we essentially saw some continuous and not very telling characteristics between these 3, but the non-melters were interesting, and we can't say that we understand it, but the non-melters essentially show this bimodal distribution, where you have a lot of proteins that have that don't melt, but they actually have not at all any disordered structure. But then there are these disordered structure proteins with 100% of it having no structure, and they also don't melt, and then anything in between. So it's hard for us to really grasp what this is, but we are just observing that this is the case. And here I showed you these 2 examples before. Both proteins are almost completely disordered. One of them doesn't crash out of solution, the other crashes out of solution rather quickly. So at this point, what we're concluding from this, and that may be not particularly satisfying, but we really looked near and far for physicochemical properties of peptides, amino acids, proteins, secondary structure, what have you. We think what nature actually has done is to optimize a protein sequence, almost like a protein design and evolution person would do, is randomize or do biotechnology on every residue until you hit the properties that you want. So it's well known that you can engineer enzymes to be thermostable by exchanging many or certain amino acids. And then you can use these enzymes, let's say in washing powder very easily and still heat up your washing machine to 95 degrees and those proteases may still work. So that's currently the interpretation, that it's careful sort of protein optimization for the purpose of making them more thermostable if they need to. Now, one snippet that we thought was interesting is when we go back to humans, we observed that there actually is a difference in the thermal stability of proteins depending on where they are. In particular, we found that proteins in the respiratory chain seem to be rather more thermal stable than other proteins. And we actually even did a functional experiment along this line, because there was a report in the literature that argued that mitochondria might actually operate at close to 50 degrees in a cell. And that we found surprising, but here at least we saw that, well, mitochondrial proteins are a little more stable, but particularly so the respiratory chain. This is actually where heat is generated, because in the mitochondria, in the respiratory chain, protons are being pumped across the mitochondrial membrane. And there's a lot of what is called proton leakage, where the protons flow back without propelling the ATPase, ATP synthase, and that annihilation of proton gradient generate heats. And so together with some mitochondrial experts here on campus, we actually did we measured oxygen consumption by mitochondria, either in intact cells or in permeabilized cells. And this is the gray and the yellow bars. And we actually found that in the permeabilized cells where glycolysis, where these mitochondria are basically independent of glycolysis, because we made holes into those cells and basically fed them with substrates for the respiratory chain, we could actually see that they would hold on to functionally using those metabolites at higher temperatures than the intact cells. Whether that means mitochondria can actually operate at 50 degrees, we don't know quite, but at least very locally in the respiratory chain, maybe these proteins are more thermal stable simply because they have to be, because the environment is warmer. I'll skip over that in the interest of being able to look at a second example. Olga, if we still have time, and I already apologize that even 45 slides are not far too many for 1.5 hours, but
we have 10 to 15 minutes.
We have 10 to 15 minutes. Okay, good. The second story that we published a while ago is about protein turnover and also proteome form turnover. That more and more turns out to be an important regulator of cellular homeostasis, and therefore people have begun to call also this proteostasis. So there are important elements of regulating the lifetime of a protein, whether you want to look at and make protein complexes or protect yourselves from misfolded or toxic proteins. This interplays with signaling. People have found that there are phosphorylation signatures on proteins that mark them for degradation. A lot of diseases are these days referred to as proteostasis or dysregulated processes like Alzheimer's disease, Parkinson's, and what have you. But also in drug research, this is becoming more and more interesting, either because these drugs interfere with proteostasis proteins themselves, like proteasome inhibitors or inhibitors of the HSP90 machinery, but also all of those covalent irreversible inhibitors that are more and more entering drug research, because once they are irreversibly inactivated, then of course the action of the drug will depend on the turnover of the time essentially when the cell will replenish that protein. So we've used TMT actually now in conjunction with SILAC labeling in what looks like a rather complex experiment, but was also sort of a data analysis nightmare for Jana Zecher, who ran that project. But it turned out to be extremely interesting to do. So we wanted to measure this protein half-life or turnover on a larger scale. And again, we are not the only ones who've done these experiments. But the twist of TMT came in extremely handy. So the classical way of measuring half-life of proteins would be a pulse chase experiment, and we did this with Silac, where you started with light labeled cells, and at some point in time you switch them to heavy, such that then all the new proteins that are made from that point forward would incorporate the heavy isotopes, and the red proteins would sort of be the old stuff that's still there. And then we basically collected samples along this time frame of the beginning to an infinite number of hours, which I think was about 5 days. And then each of those time points were encoded by TMT labels, and we then mixed them all together and then did some fractionation and then LCMS analysis on this. And it turned out by the times we chose that we usually got Silac pairs that were roughly 1-to-1, because it depends, of course, on the doubling time of those cells, what that exact time frame here is. But then we asked the mass spectrometer to both fragment the old proteins and the new proteins, and that from MS2 spectra we would then pick a couple of those fragment ions to do an MS3 experiment where we would detect the TMT reporters. And that enables something rather interesting, because now you can plot those TMT intensities of the old proteins over time, and you can fit curves, degradation curves to them that tell you how quickly those proteins disappear from the cells because of degradation. And conversely, the blue channels, or the newly made proteins, we could now generate synthesis rate curves for each of those. And that is important to use TMT in an MS3 mode, because if you want to measure synthesis, really your time zero must have 0 value for that peptide. And if you only do this in MS2, I can tell you from experience that most of the curves start at 0.2 or even 0.3. So then when you fit these curves, you're actually getting quite wrong data. So here the combination of Silac and TMT allowed us to get these degradation and synthesis curves in the same experiment. And from that, essentially for each protein, ignore the stuff on the left-hand side. There's just a lot of quality control. But that allowed us to generate synthesis and degradation curves for a lot of proteins. And you see 4 lines here, which are 4 replicates, and 4 lines here are the same 4 replicates. And of course, the crossing point here always would tell you something about when you have 50-50. But then you have to correct this for the doubling time of those cells. And then you can compute, for example, for this cyclin protein here, that it has a half-life of about 3 hours, whereas this ribosomal protein has a half-life of about 100 hours. And when you plot that out for all of the proteins that we've got, then you can see that on average, the half-life of these proteins is about 1.5 days. But then there are also some that have really short-lived and others that are very, very long-lived. That to some degree correlates with the copy numbers per cell. So the more abundant the protein is, the longer it tends to live. That makes, of course, some sense. Again, when we ask the question, do longer proteins, are they less stable or have they shorter half-lives? The answer is no. And again, when we turn to more structural elements, we saw actually that the polar amino acid and coiled-like structure had a destabilizing effect, and then hydrophobic amino acids had a rather stabilizing effect, akin to what we actually saw in the temperature experiments in my first study case. Okay, we also broke this down into parts of the cell, or what they do. Here in parts of the cell, you can and just rank them by their half-lives. This is now in log 10 scale. Here being on the left-hand side being the most unstable ones, on the right-hand side the most stable ones. Again, very hard to make general statements about it. You can see perhaps a trend along this if you want to order it this way, but it's very hard to really conclude on a protein by protein basis anything from where they are in the cell. What was more telling is actually an enrichment analysis of the half-lives, where we saw that many people already saw before, is that particularly transcription factors tend to be short-lived, which of course makes sense for the cell if it has to respond to some changing situation. It either wants to make more proteins quickly, or it wants to stop making certain proteins quickly, and the transcription factors would be one way of doing this, whereas the more stable proteins also tend to be more enriched into processes that the cells need all the time. Again, here coming back to the respiratory chain, we also again saw something that we already saw with the thermal stability. We looked at which proteins would be actually more stable or less stable, and we actually found that proteins from the complex 1 of the respiratory chain tended to be rather unstable, and we wondered if that is because of oxidative damage. And if we look at here the respiratory chain over here, in complex 1, this is where also a lot of oxidative stress actually happens, and the half-life of the proteins in complex 1, at least on average, were this is again a log 10 scale, quite a bit shorter than that of others. And basically the point of this plot down here is when we map this out onto the structures of these proteins, we see it really only affects proteins that are in the lumen and not the proteins that are in the membrane. And one potential explanation for the lower or higher turnover of these subunits is that they potentially accumulate oxidative damage and therefore potentially lose their function, so they have to be exchanged more often than those proteins that sort of sit in the membrane are not exposed to as much oxidative stress. So in the last 5 minutes, I would like to quickly go through, again, coming back to something computational, if I may, where TMT is also beginning to play a role. And some of you may have heard about our Proteome Tools project, where we've some time ago started making a lot of synthetic peptides with the aim to represent them as a human proteome in terms of synthetic peptides, and essentially to create sort of a ground truth data set for LC-MS-based proteomics, and then build prediction tools from the tandem mass spectra that might work for any protein from any organism. And that kept us busy for some time, but it was and actually is a very fun project where we made all these peptides and fragmented them in a lot of ways and got hundreds of millions of PSMs, and the data is all there for people to use. And initially we did this all label-free, but we've then added a TMT component to this to make, for example, spectral libraries that are available from various places. And just a few snippets for why we actually wanted to do this. Much of it was driven by the wish to have better error control in proteomic studies other than using classical target decoy type of error control methodology, because we saw that they all have their limitations and that can accumulate in very large data sets. So it's quite clear why this may happen. For example, you have here a peptide or an experimental spectrum here in point of the signals pointing up that Andromeda matched to a sequence like this. And then the second best match to that very same spectrum is a peptide with a rather different sequence, and the score is almost the same. It's hard to distinguish for the search engine because the search engine in this case kind of doesn't really know what intensities to expect from those peaks. Whereas when you make synthetic peptides for exactly this peptide and record their spectra, you can see from a mile away that this sequence, this synthetic peptide spectrum, looks almost identical to the one that Andromeda has matched as a second best, and the best one actually is something completely different. And so we can clearly rule out this one and say this is the much better match. And we wanted to do this systematically, and because there was a lot of data there, Sigi Gesulat trained a neural network on this data to basically try to predict the fragment ion intensities and also the retention times of all these peptides. That turned out to work extremely well, and that made us extremely happy, because at the beginning it took us a lot of time to find a funding agency that would give us some money, because everybody said, why do you want to do something as boring as make a lot of synthetic peptides? And we are very happy that we did, because it turned out that this prediction tool works rather well. And in this plot here, it may be a little complicated to understand at first, but essentially what it shows you is a spectrum of a peptide of this sequence. And in red are the Y ions and in blue are the B ions. And if you see a lot of red and blue, that means that the actual experimental spectrum and the prediction made by that machine learning tool agreed extremely well. And if there was a little bit of green, then there was a little more signal intensity in the experimental spectrum. And if there was more yellow, then the prediction would have thought there should be a little more intensity here, but we didn't see that. But when we looked across many of those spectra, we saw that this tool that we built that we call Prosit did rather well when we compare experimental spectrum to predictions. So in Spearman in Pearson correlation world, that would be north of 0.99. In spectral angle world, it's still better than 0.9. And that has enabled or is enabling quite a few things. One is, for example, rescoring data where large search spaces are a problem. And one example is here shown for metaproteomics, but it would also be true for HLA peptides or for doing proteogenomics, where you have to consider a lot of sequences, and therefore your error control becomes harder and harder just because your sequence database against which you're searching becomes bigger and bigger. And I would like you just to look at this part of the figure. When we search a metaproteomic data set that also contains human data against just the human database, then we'll, let's say in this case, find about 3,000 peptides. If we make the database bigger by asking, well, let's search against the human sequences of UniProt and all bacteria, we all of a sudden begin losing identifications we had before. And we also gain a lot of identifications of bacterial peptides that we didn't have before. But then when we make the search space even bigger, which would be that huge green blob down here, Andromeda really finds it very hard to find anything. Now, if we then basically ask Prosit to predict all these spectra and ask now the question, how well do the experimental and the predicted spectra match to each other, we can actually see that at 1% false discovery rate, we all of a sudden get 50,000 bacterial peptides. And yes, we're also losing human peptides, but not quite as many. And we can also crank this FDR up to 0.1% without really taking a big hit. So we think that all these spectra are very useful for this type of rescoring exercises. And the reason why this actually works is, that it's better able to discriminate false and true matches. Over the past few months, we have also sort of extended this to non-tryptic peptides or modified peptides, and it seems to be working quite well because a lot of these peptides that we've made are also modified. And here's an example where phosphorylation site localization may be improved. And I'm just going over this reasonably quickly because I wanted to come to the TMT part, which is sort of the latest addition. So we've also now trained Prosit on TMT labeled peptides. And you can see these mirror spectra up here where we have this particular peptide that is TMT labeled, and the peaks pointing down are the actual experimental spectrum, and the peaks pointing up is the prediction. And again, and comparing and contrasting that to the non-TMT labeled version of the same peptide, which actually does look different to this spectrum. But when we look at the violin plots, the story is the same for as for the typical unmodified peptides and non-labeled peptides, that it seems that Prosit is learning what these spectra should look like. And therefore, once we are able to release this, we hope that people find good use for this. And with this, really, I'm coming to an end of the presentation. I apologize for taking a lot of time, and I hope I didn't bulldoze over too many of you with too much information. But of course, we have now some more time for questions, I guess. And I would like you also to just take a moment, if you have, to look at Proteomics DB, where many of the things I talked about eventually end up in, either be it large data sets or be it computational tools. And Proteomics DB can either be explored with your browser or with APIs for the computationally inclined people. And we hope that this continues to serve the community well. And with that, I really have to say thank you to a lot of people that have done all the work, and also a lot of collaborators around what I presented today, around be it the Meltome Atlas or be it creating Proteomics DB or creating Proteome Tools and Prosit. So with that, I say thank you for all of you who might still be here. I have no idea how many of you are still here because I don't see how many people are in the presentation, but it doesn't matter. So thank you for all of you who stayed until the end. And I'm happy to take more questions.
Thank you so very much. We have a round of virtual applause from over 450 participants who were listening to your presentation. So we have time, we have questions coming up more. So let me just ask one question, which kind of maybe wraps this up from one of the participants. So what is your current decision tree for proteomics experiments? So how do you decide when you go with LFQ versus TMT?
Yeah, so we really very carefully define the set of questions we want to answer right at the beginning. And to the extent possible, we're going to make that decision rational. Sometimes it has to be practical. For focused small experiments, we actually tend to do more and more and more TMT, because it allows us to put replicates together and directly compare experimental conditions. And we have taken the decision in the lab that whatever we would do with TMT, it's always going to be MS3, because we want to capture some of that dynamic range and accuracy that we can only get from MS3. We know we get less data. We get fewer peptides, fewer proteins, but the quality of that quantification is much better. So every time we do TMT, in the end, it's always going to be MS3. Most of the sort of largest experiment, and let's say for the melting experiment, there was really no other choice. You can't do that many experiments in a label-free fashion, because it would be 10 times more mass spec time. It's just completely impossible. And it's not that my lab has 20 instruments. No, it's not even that my lab has 10 instruments. So we have to also be mindful of these practical issues. But I think the short answer is we really try to operate a fit for purpose approach every time we come up with a study or project.
Well, thank you so very much. So we have a lot more questions. So I'm carrying the questions over to the Google Doc. And Bernhard, if you don't mind, take a look at the Google Doc and see if you can give answers. You know, there are many questions, so to the extent of your ability, just answer a little bit. And maybe let me also mention two things. So tomorrow and Friday, we have 2 days dedicated to work with Skyline. And it is my understanding that Brandon, who is the developer of Skyline and the leader of the 2 sessions, will be talking about integrating Prosit with Skyline. So
yes, I forgot that. That was actually a fun summer intern project that we got Prosit into Skyline. So we're very happy about that. Yeah.
And another plugin is that next week, I think it is Thursday next week, we will be talking more about statistical analysis, and we will have a presentation dedicated to MS.TMT. So there were many questions about normalization, reference channels, things like that. So we will certainly be discussing this in the context of MS stats.
Okay.
So thank you, Bernhard, once again. So it is wonderful to hear from you. And I think it will be a very, very useful resource for many people who watched now, but also who haven't had a chance to join us. So it will be available on YouTube. So you can share this with your group members. You can share this with your colleagues. So we're a little bit over time.